Okay, let's talk about Chapter 16, the nature of communities. The key concepts for this chapter are that communities are groups of interacting species that occur at the same place and time, that species diversity and species composition are important descriptors of community structure, and that communities can be characterized by complex networks of direct and indirect interactions that vary in strength and direction. The chapter starts off with this example of an introduced algae, and the method for introduction was actually an aquarium release, which is not that uncommon, actually. Most aquaria have better practices now, but for quite a while, excess animals were just tossed in the water and assumed not to survive. In fact, when I think it was Marineland, a SeaWorld-esque park went out of business. They had all these fish left over. They sold the valuable ones or gave them away, marine mammals, etc. But, they had like a lot of tropical fish. This is off, I can't remember if it was Palos Verdes or closer in San Diego, but Southern California. They had a bunch of animals, fish that they didn't need and nobody seemed to want. So they just opened the drain and dumped them all out. And luckily none of those stuck. There's another example actually in the Great Lakes where there's a salmon hatchery and they were rearing pink salmon or humpies. And they were actually rearing them for introduction into the east coast of I believe it was Canada. And the goal was to replace the eel populations that had been declining for a number of First Nations groups. And so they basically had screwed up the eel population. And so they were going to plant Pacific salmon in those rivers to take over. And they gave them pink salmon, which is the crappiest of the salmon. And they never took. And so they kept putting pink salmon fry in these East Coast rivers, and then never took. And the hatcheries in the Great Lakes region. So they just pulled the plug and dumped all the pink salmon fry in the Great Lakes and thought that, well, whatever, they're all, you might as well go to fish food, right, instead of just throwing them away. Well, it turned out they survived in the Great Lakes, and so now there's a pink salmon population in the Great Lakes that was due to an accidental a discharge of dumping animals into a body of water which they're not native in. So similar to that, those wound up taking hold, and similar to that, this algae is taking hold, where it normally lives in a much warmer climate, so they didn't think they would survive in the Mediterranean. But turns out, this algae species had been in aquaria for quite a while and had the ability to survive in this colder environment. Really takes over and kind of forms this monocrop of algae. And then the question is, like, what does it do? Like, how is it impacting other native species? And there's been a couple removal programs, but trying to remove marine algae once it takes place is pretty difficult. But this did give the opportunity to track the distribution and spreading of the species. And so it was first released or first found off Monaco, and then has since been observed in other areas, likely transported via anchors, other fishing gear, things like that. And once it's established, it's pretty hard to get rid of. In the aquaculture project I'm a part of, we went to Hawaii in February 2020, and that was one of the big things that they're working with fish ponds, is all these invasive algae in Hawaii. And basically, it's just like maintenance. They just have to go in and they have this giant vacuum. They go through and vacuum up all this invasive algae. They're never going to get rid of it, but it's, they can try to maintain it effectively. Is what they're doing through physical removal, but it's never going to go away. It's always going to be there. Once an invasive takes hold to that extent, extirpation is nearly impossible. And so this Caulerpa started from the Oceanographic Museum in Monaco, and it turned out to be a cold-resistant strain that had been sent to a zoo in Germany for tropical fish displays. It was released in the process of cleaning tanks. The workers thought it wouldn't survive in the cold Mediterranean. It turned out not to be the case. And so based on the genetics, they can track exactly where it came from. And that's pretty interesting. And so far, we've considered species interactions in a two-way relationships, predator-prey, positive, negative interactions. But in reality, species experience multiple interactions that shape communities in which they live. And so we'll start to kind of layer on natures to these, layer on interactions to thinking about how ecology works. And particularly when we talk about the ecology of the environment, we're typically talking about within a community. And a community is one of those squishy terms like, well, what is a community? Is a community for bluefin tuna, like the Atlantic Ocean? Or how are we defining that community? And we typically use either physical or biological characteristics and generally scaled to the organism we're looking at. And so we're not looking at the entire distribution of the species, but some small population of that species within a certain area, and we call that area a community. And so we can define that either physically or biologically. And so physically, if we're looking at a hot springs, that's pretty simple. It's like there is a hot spring. It is a discrete space, or perhaps a desert ecosystem, over some scale of perhaps tens or hundreds of kilometers. We can also think about biologically defined communities, things like a kelp forest. And there we will typically look at one kelp forest. And so that would be like the kelp forest off of, I'm trying to think of a good local example. There's a fair amount of kelp off of the west side of San Juan Island, and there's a fair amount off, Shark Reef off the southwest side of Lopez. So that would be like a patch of kelp. And that patch of kelp might be a kilometer long. It's likely around here smaller than that. If we're looking for the largest kelp patch in the world, it would be off Point Loma, and that would be maybe a couple kilometers long. But generally a discrete patch. And there would be a kelp forest then surrounded by a non-kelp environment, either too shallow, too deep, or if you're thinking about that way, left and right, not suitable habitat. Coral is the same kind of thing. You can zoom out and think about, well, like all of the coral, in the Caribbean, or you could think about a patch of coral off Plow, or I'm trying to think, what's that island that everybody studies? I'm blanking on it now, but you can think of like a discrete patch of coral then surrounded by sand, and so that would be your community in that case. And so our ecological work is typically in communities, and that's also the scale of the kind of question we're asking. What's the role of herbivory in a kelp forest? We would look at discrete kelp patches that we can follow through time and monitor herbivores within them. And we would use that community and then extrapolate out to other kelp forest ecosystems. And so the idea is to study a community and then derive some conclusions from that study and be able to relate that to other communities that may be near or far. And so as ecologists, we often define a community arbitrarily based on the questions you're posing. An example could be the study of aquatic insects and their amphibian predators might restrict the community to that interaction and exclude things like birds, which could be migratory birds that would really, go from Central America all the way up through Alaska, right? And so we're really looking at what are the parts of the ecosystem we're trying to tease apart, and then what's the scale we need to understand that interaction or mechanism, and then define our community around that. And so counting all the species in a community is essentially impossible, particularly when we get to small things, when we start to include the bacterial or viral communities. it gets hard to just go through and count everything, are small cryptic fauna. So ecologists usually consider a subset of species when they define and study communities. Both allows us to look at just a few organisms that we think are important to that interaction, and then be able to collect meaningful data. If we have to count every single thing every time we go out, it gets to be quite difficult. And we can also kind of group things to make it a bit easier. We could have taxonomic groups, i.e. all bird species or all snails. We could put all gastropods together, all bivalves together, and just kind of say, okay, here's what's happening in the bivalve community. And that might be important if we're looking at predation, where like red rock crab or sea stars might not only be eating one or two types of clams, they could be eating a broad suite of clams. And so we might just, bin all of the clams together and call that a taxonomic group of bivalves. Or we could look at guilds, which is a group of species that use the same resource. So you can have, like on the intertidal, you can have barnacles and what's called a porcelain crab. And a porcelain crab is a small intertidal to subtidal shore crab that looks different than most of the shore crabs you see when you roll a rock over. Instead, these typically have big, kind of flat pinchers, and they're actually filter feeders as well. And barnacles are filter feeders. And so while I guess they're both crustaceans, but while they're, pretty different, they're still consuming the same set of resources. And so you would assume that if water flow is good for barnacles, it would also be good for porcelain crabs because they're also filter feeders. And then you can look at functional groups, species that function in similar ways. And we'll look at the example given for that. So taxonomic affinity, group of birds. A guild could be pollinators or nectin consumers. And a functional group here are groups that all fix nitrogen. And so it could be a wide variety of plants, but they all have nitrogen-fixing bacteria. And so therefore they're all kind of doing the same function. So it might not matter what species the plant is, the function is still the same. It's still adding nitrogen to the soil. So we can kind of think of them in that way as far as like maybe ecosystem service would be one way of thinking about a type of functional group. And then we can put things in food webs. And this is a way of organizing species based on energy flow. And so we're really looking at where does the energy move through the system. And we have to start with our primary producers, autotrophs. We typically jump to things with chlorophyll, although it can be chemosynthetic bacteria or archaea. But generally, most of our systems, the primary producers are photosynthesizers. But again, it can be other sources as well. Then you have primary consumers, herbivores. They eat primary producers. So these are the things eating, again, primarily plants. And then secondary consumers that are eating the primary consumers, and then tertiary, which are carnivores eating the secondary consumers. There's few examples beyond that. Typically, they'd be in the marine realm. You can get higher than this, but it's pretty rare. So if you think about an orca whale, an orca could be eating a sea lion that's eating a salmon that's eating an anchovy that's eating really a combination of zooplankton, sometimes large phytoplankton. It depends on the species. So there we can start to build out additional trophic levels. On land, it's a lot harder. So if you think about a grassland and you've got deer on that grassland, herbivores, and you've got wolves that are eating those herbivores, what eats a wolf? Not that many things. And so on land, really to get to a tertiary, you could think about that wolf could be eating maybe like a muskrat that that eats, I don't know what they eat on land. Fish, I guess. But here we got like river otters that eat crabs and things like that in the marine system. So a small carnivore. But really, to get beyond that, it gets pretty difficult in the terrestrial realm. In the marine realm, we can start to get to fourth or even fifth level consumers. Again, or a great white shark that's eating, again, like a stellar sea lion that's eating a salmon, that's eating a fish that's eating zooplankton. The zooplankton are eating phytoplankton. So there we'd have phytoplankton as our primary producers, zooplankton as our primary consumers. We'd have herring as our secondary consumers. We'd have salmon as our tertiary consumers. And we'd have stellar sea lions as our fourth consumer, and then our fifth level consumer being a great white or perhaps an orca whale. So you can do that in the marine environment. It's very rare on the terrestrial environment. Thing like what eats a grizzly bear, or what eats a lion, or what eats a wolf, or things like that. We don't really have that much because space becomes a big issue where in water organisms can move around, and they're also your primary producers are smaller. Phytoplankton are smaller than grass, and so you're kind of starting at a smaller level. So it's interesting that the different number of trophic levels between marine systems and terrestrial systems. And so we can look at the food web here with our primary producers, autotrophs. They'll be consumed by herbivores. We can have omnivores that are eating both herbivores and they're eating some of the primary producers directly. We can have a number of herbivores consuming the autotroph. They're being consumed by the second level or secondary consumers, and our tertiary consumers that might be eating a wide variety of secondary consumers. And again, back to that idea of typically carnivorous organisms are more opportunistic, that they typically don't hone in on just eating one type of prey because it's so energetically expensive to chase around animals that you tend to eat whatever fits in your mouth, compared to an herbivore that might be able to specialize, but either in time and space with a single type of again, we use the word prey when we're talking about a herbivore eating a plant. It's kind of funny, but we tend to use that. So whatever their food source is, they're able to specialize in that. But what food webs don't tell us is the strength of interactions or the importance to the community. Some species can span multiple trophic levels, omnivores, or they might change what they eat through their life cycle. Frogs is an example of a tadpole that could be herbivorous in tadpole stage, and then when it metamorphoses into a frog, it might be a carnivore. And so through its life, it's an omnivore. Any one given time, it's either eating autotrophs or it's eating animals. Other things will eat both at the same time. Bears are an example of an omnivore. They'll eat berries or they'll eat fish or other animals. And many of these food webs rarely include symbionts and detritivores as well. So we think of this like food web or it's like an apex predator, but we're forgetting what happens when the apex predator dies. They're still food for somebody else. And food webs do not include non-trophic interactions, horizontal interactions, competition, positive interactions between species at that same level. Meaning, what happens if you have 10 different types of herbivores out on a field? What kind of interaction are you going to have that's important to that species and the outcome of that species, whether it be survival, growth, or reproduction? Because now we're introduced to competition or perhaps some of the positive interactions we talked about in the last chapter. And so interaction webs can be added to include some of these vertical trophic, so vertical up and down, and horizontal non-trophic interactions. So we can start to think about the prior food web now with some interactions between them and the connections between them. So here the horizontal areas can be competitive or positive interactions between the two. So presumably if you have two herbivores consuming the same primary producer, having more species on the landscape would reduce food resources for either one of them. Having two secondary consumers here may be a positive interaction where maybe the tertiary consumer will favor one of those, and so if you have a weaker tertiary carnivore in the system, then maybe that's good for you because maybe that tertiary carnivore is easier for the the secondary consumer, that other carnivore, maybe that's easier for your tertiary consumer to consume. And so having another species there, you might think of as a negative, but if they're slower, maybe that's good. Maybe those will fill up on those instead of your species. So adding those interactions helps us think through, these ecosystems. We're trying to model them, and we're trying to model them not necessarily to replicate nature, but to be able to ask simple questions. What happens if we have more or less of this? And really the interaction we talked about before with orca whales, otters, urchins, and kelp is really, it's a food web interaction, right? And so when you add one, what does it do to the subsequent layers? And so looking at food webs or interaction webs are ways of thinking about that. It's ways of producing a model of the environment so we can both ask simple questions and then put our results into some sort of context, kind of adding a mechanism to what we're seeing. And so community structure is a set of characteristics that shape communities. It provides the basis for generating hypotheses and experiments to understand the communities we work. And species diversity is a common measure of community structure. And so, who's there, how are they interacting, and then how do we measure changes in that ecosystem? And species diversity is a good way of doing that. There's many different ways of looking at species diversity. One, we can look at species diversity, and we can talk about species richness, which is just the number of species. Just going out, and if you can imagine throwing a quadrat in your lawn and going and counting up all the species. That's your species richness. That's the number of organisms there. Or you could look at species evenness, which is a relative abundance. And so if you find, I'm going to show my ignorance around lawn care here, but if you find clover, a dandelion, and a bunch of Kentucky bluegrass, what's the relative abundance of those species? You would assume that your Kentucky bluegrass is very high, maybe clover, and maybe you've got one or two dandelions, and so you'll have a ranked level of abundance. So you could have 3 species in multiple quadrats, but the species evenness might change depending if you land in like a patch of clover or a patch of dandelions, and you've got less of your grass species. Kentucky bluegrass is a type of grass. I have no idea if it's what we grow here. I don't know. In the example, both communities have the same species richness, and community A is dominated by one species. This is referencing down here. Okay, sorry, I'll jump to the next slide here. So we've got 2 communities. They have the same number of species. So when we're looking at species richness, it's the same. Both have 4 species indicated by the different colors of butterflies. So we have a species richness of 4. Here we've got 1, 2, 3, 4 species, same species richness. However, species evenness is totally different between community A and community B. Community A is dominated by, I don't know, let's call it the purple one, where community B is more even. So here we've got a whole bunch of, again, I'll call it purple, I don't know if it's purple, and then one member of these 3 species, Where here we have a more even distribution. So we've got 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. So here we've got 4 species, 5 of 5 members of each species. So we have a completely even distribution of species. Where here we have the same species richness, i.e. the same number of species, but the relative abundance is totally different. A whole bunch of one species and the other species are very rare. So the reason we look at richness or evenness as well is because the richness doesn't tell the whole story of it, right? If you're concerned about loss of species, having small numbers is how you can lead to local loss of species. And so if we have a very even system, then we could, there's some resilience built in, there's some strength built in for those species to resist loss. Where if we have low species evenness, these species are all at risk. There's not that many of them. And if we want to combine this and look at an index we can measure and compare between sites or through time, one of the most common ones is called the Shannon index, and we'll walk through this. The equation looks a bit scary, but we'll walk through it, and hopefully it makes sense. And so all we're doing is there's a negative sign here, so we'll come back to that. But it's the sum of the proportion of individuals for a given species times the natural log of that proportion of abundance, and we add that up for every species in the system and then times it by -1. And so we'll walk through an example here. So here, I guess they were blue. I thought they were purple. I apologize. So here is the calculation for species diversity for the butterfly graph. And so we'll talk through this and we'll jump back and look at the butterfly data. So we have our 4 species of butterfly: blue, yellow, pink, and orange, and then the total. In community A, we have 17 blue, 1 of the other 3 species. And so the relative proportion abundance is 8.5. And so that would just be 17 divided by 20. So the number of that of individuals for that species divided by the total number counted. And then for yellow, pink, and orange, it would be 0.5, so 1 divided by 20. So that's abundance and proportion. Go down here and look at community B, where we have an even distribution, where we have 4 species, blue, yellow, pink, and orange. There's 5 each, and then the proportion of that's 0.25. So the 4 species each represent a quarter of the overall number of organisms we count. So you can think of proportion as the number of organisms represented by that one given species. So I'll jump back here and we can look at it again. So here we've got the same richness, right? We still have 4 species in community A and community B. Community A, it's dominated by the blue butterfly. 17 blue butterflies, 1, 2, 3 of 3 species and 1 of each. For community B, we've got a very even distribution. We have 4 species and 5 of each member. So that's how we come up with abundance, and that's how we come up with the proportion. Now we're taking the natural log of the proportion, so here we've got those values, and then this is the proportion times the natural log of that proportion, giving you these values. And then we sum up all those values, we just add them up, and now we've got a negative number here, negative 0.5 for our uneven community A. For community B, we have the same number of species but a more even distribution. So here we're multiplying 0.25 by the natural log of 0.25 to get negative 0.3. We add that up, and now we have negative 1.3. Now we'll go back and remember that we're multiplying this whole thing by -1. So our H value, which is the Shannon index value, our Shannon index value is not negative 0.5, it's actually 0.5, and here it's not negative 1.3, it's 1.3. And so here, for a given number of species, a given species richness, the H index, the Shannon index, is higher in a more evenly distributed system. So if we have the same species richness, but higher species evenness, that'll give us a higher H, a higher Shannon index. Now, because we're just summing this up, if we had 10 species here, we'd start to get a larger value here as well. So the H index increases with species richness, the number of species we're adding up, and species evenness. So it's a way of combining both of those into a single metric. And so we can look at H indices, or the H index, over space or time. And so if we wanted to go back to the cleaner shrimp example in coral reefs, removing cleaner shrimp then decreases species abundance, and perhaps impacts evenness as well. And so we could look at the H index of a coral system with cleaner shrimp and the H index of an ecosystem without cleaner shrimp, and compare that impact on the overall species diversity, species richness and species evenness. So that'd be one way we would use the Shannon index. Species diversity and biodiversity are often used broadly to mean the number of species in a community. However, biodiversity can be used to describe diversity at multiple scales, and so from genes to species in the communities. And so when we really define biodiversity, it's important to understand it includes genetic diversity as well. So if we go from a species that's distributed all around the nation to it only surviving in a small pocket, and that species going through a genetic bottleneck, fixing a number of alleles and losing some other ones, that is a loss of biodiversity. Now, we could still have that species, it still exists, but it's lost biodiversity in that it's lost genetic diversity. And obviously genetic diversity is important for the survivorship of the species, its ability to adapt as environments change, its ability to reduce inbreeding impacts, things like that. So when we talk about biodiversity, it's not just the number of critters on the landscape, but it's also the genetic fitness of those organisms. And so here's one scale of looking at biodiversity where we could have, the same species of moth, but with a variety of different genes that relate to size or coloration. And so including that into our biodiversity, it lets us know how this butterfly can adapt to changes in coloration, changes in seasonal patterns, things like that. So we can measure diversity at multiple scales, both just the broad term of like, this is a blue butterfly, or this is a blue butterfly with a large degree of genetic variation. So rank abundance curves are a way to plot the proportion abundance of species relative to others in rank order. So this is another way of kind of looking at species distributions and kind of evenness. So this suggests possible species interactions. In community A, the dominant species might have a strong negative effect on the 3 rare species. And you can add experiments to explore these relationships. So if we look at, back to the butterfly example, if we just plot the proportional abundance for the most dominant species, that's the first one, it's quite high, and then for the last 3, it's low. It's 1 individual out of 20. Where community B, it's very even. So I'll go back to the figures and we can look at this again. So there's a way of telling you that, maybe if this one's lost, there'll be a big change here. It's a way of like asking that question. Okay, there's something going on in community A where the blue butterflies are dominating, where in community B, they're all in equal abundance. And so what's the mechanism for that? What's happening here? And without observations, then we can start to derive questions about that, whether it be food source, competition, predation, something like that. So we'll go back and look at these numbers are coming from the relative proportion here. So 8.5, 0.85 for the blue and 0.5 for the three others. And again, an even, a 0.25 relative species proportion for the 4 species in community B. So again, even, high species evenness, they're all, their relative abundance is all the same, or community A is dominated by the blue. So we can remember those figures. Community A, if we're comparing butterflies in 2 adjacent valleys, and we find this this proportional abundance for community A is a lot different than community B. There's, that's a foundation of a good research question of like, what's going on here? Why is that happening? We can also look at species accumulation curves. That species richness plot is a function of total number of individuals counted. And this is widely used because the number of species you count is directly relative to how much effort you put in. If I'm trying to count insects in a stream, and I go and I throw my quadrat and it lands on a stream, and I count 10 species, and okay, I'm good, I'm going to go home. Well, what would have happened if I would have thrown that quadrat again and again and again and again? How many times do I need to throw my quadrat before I count every species in that ecosystem? Particularly when we're thinking about rare species, it might be a lot. And so it's good to know, basically it's like that trade-off between effort and data. Like, where is that starting to plateau out? Where if I sample 1 or 2 or 3 or 4 more quadrats, I might add 1 or 2 more species, but my first couple quadrats, every time I threw it, I would find 10 new species. So eventually there's kind of a reduction of rewards there. And so, how much is good enough? And so this is a way of looking at sampling effort versus the number of species in the samples. And so we go out and take our first sample. It's our first sample, so every species we find is a new one. We go out and take our second sample. Same thing, there's lots of species here. So our second sample has a whole bunch of new species. Third, fourth, fifth, and it kind of starts to plateau off. And so after a certain amount of sampling effort, we just start seeing the same things over and over again. And every once in a while, we might pick up a new species. And so we want to sample until we get, to start to approach the asymptote here. If we stop sampling here on the exponential growth part of the curve, we don't know if this keeps shooting straight up or where it starts to plateau. And so in reality, the threshold for no new species are counted occurs in natural systems because new species are constantly being found. And so in theory, if we had infinite sampling effort, we would start to find many more species. But in reality, our resources are not infinite. So Hughes et al. Looked at this in a few different areas to kind of compare different relationships between sampling effort and the number of species found. And it was kind of an interesting one because they included like really different communities. And so they looked at temperate forest and tropical birds, and they think they adequately represented the number of species of bird species was adequately represented before half the individuals were counted. They looked at tropical soil bacteria, and more effort was needed to sample this immensely diverse community because we're looking at bacteria. They also included human oral mouths as well. So at tropical soil bacteria is this line here. For every unit effort, they got more species. And so particularly when you're looking at microbes or bacteria, generally the more you sequence, the more you find. And so here there's just so many different types of soil bacteria that they haven't even, started to slow down in the number of species they find. So every sample has the same number of new samples. And for tropical moths and human oral bacteria, there was a steady incline, then it started to decline, but still not really at the asymptote here. It's not really not leveling off. Or for birds, quick incline, and then it took more effort to start to get new species. And so we can plot this one out and predict where it's going to end. Here it's a pretty big guess, and in here there's like no idea how, this is not nearly enough samples to decide, do we need to, can we predict how many are there based on the shape of the curve? Or here we can fit a line to this and say, okay, presume if we sampled 1,000 more times, here's how many species are going to be there. And so scale is important. If tropical soil bacteria were sampled at the same scale as Costa Rican moths, the bacterial diversity would be immense in comparison. Meaning if we sampled, 10 kilometers by 10 kilometers, we'd find a massive amount of bacterial diversity. Where if we look at the bacterial diversity in a drop of water, that's a lot easier. And the study highlights how little we know about community structure of rarely studied assemblages, microbes, etc. Community composition is important. The identity of species in a community. Two species could have identical species diversity values, but completely different species. And the identity of species is critical to understanding the community structure. So measuring, Shannon diversity index is really useful, particularly if you're comparing the similar suite of organisms through time or space, but comparing two entirely different ecosystems, deep sea versus tropical forest. Maybe it's maybe we need to think about the species composition, who's actually there as well. So I'll wrap up the first part for lecture 16, and I'll come back and do the second one.